like physical change in the landscape. You might think as well about changes in the chemical environment, the hormonal changes of pregnancy, and how that might reshape the landscape at the time of either rebuilding a microbiome during pregnancy or the subsequent transferal to a baby, and then biotic, the actual species and their arrival. Again, there is growing work, bodies of work, to suggest that these are not just concepts that sound plausible and relevant, but in fact can be demonstrated to be important experimentally. And this is a paper I won't review, but simply to highlight, recently published, Andrew McPherson and his colleagues in Bern, Switzerland, in an elegant series of experiments show that a mouse, a germ-free mouse, when pregnant and then transiently exposed to microbial species, transient in the way that they then disappear before delivery, that the subsequent pup, this is a mouse pup, now has a different looking immune system, a different trajectory of immune system development, and a whole set of different associated properties, physiologic properties, than would a mouse that was born to a germ-free mom that had none of those intrapartum during pregnancy exposures. So we need to be thinking now about this history and its impact on fetal development, fetal health. We know about epigenetic imprinting. I would submit that the presence of an evolving microbiome during pregnancy is another form of both direct and indirect epigenetic imprinting. It's now going to be transferred to a baby and play out over subsequent years. And this idea of playing out over time is something, again, that's been hypothesized to be important, the idea of serial successions of community propagation and assembly. This, again, is not just a concept, but it's been shown in experimental work, again, now here at Stanford by Justin Sonnenberg, in a really interesting series of experiments where he altered the diet of a first-generation set of pregnant mice, and then looked at subsequent generations of mouse pups born to a mother that had been put back on an originally, in this case, fiber-deficient diet, and saw that it was only over successive generations that one saw the persistence of an effect on these growing intergenerational mouse pup microbiomes that he then concluded was the result of this history that had now been propagated out over time, now over generations. So, what about the effects of pregnancy on the microbiome? There are some data in the literature. We saw it here in work that was supported by the Marcia Grimes Foundation and through a prematurity research center that I think you may have heard about, hosted and guided by some of the people in this room. We saw it as part of a microbiome-oriented component to look at what happens to this microbiome during the course of pregnancy. And I won't belabor the details. This is published work, that which I will show here. But in short, we looked at a variety of body sites in a mother, looked at this longitudinally over time, and asked the mother to sample once a week during the course of pregnancy, although this begins, generally speaking, only after 12, 14, 16 weeks of gestation, because we had the challenge of trying to get to women who were now pregnant and willing to join our study. And I'll tell you about some of the limitations at the end. So, this was sort of the setup, weekly sampling, these little green bars, multiple body sites, lots of samples, collection of mother host-related samples as well. Well, for the purpose of host-related analyses, everything is egg host and environment. And I'll tell you about, again, the data that were published for a set of 49 subjects. We started with a preliminary set of 40 and then sought to validate the findings in a subsequent 9. I will simply say that these were women recruited from the Palo Alto area, so they had reasonably good representation within the Hispanic ethnic class, but limited representation within various racial classes, including African Americans. And the women who had delivered prematurely, which in fact, is one of the major goals of this research center, to understand the basis for prematurity. These women in our study delivered generally a bit late for a premature delivery. So again, one of the limitations was the limited number of subjects that we had, their restricted racial background, and the numbers of women in particular who had delivered prematurely, given that this was one of our primary goals to understand. So lots of samples over time. These are the 4 body sites. Each of those little rows is a woman, and the dots are the samples. You can see the collection. This research center, and all of its wonderful staff, continued the collection of samples after delivery, and I'll say a little bit about one slide those data as well. This is a display using a form of ordination, of course, premised on the idea that we have multidimensional data, and we're seeking to visualize it in a way that we can begin to build hypotheses. These 2 axes here represent the greatest amount of variation in these data, and these data are the taxonomic composition of the samples from each of these 4 body sites. And in this display, those samples are color-coded based upon the body site from which they were derived. And these various 40 women now, this is the initial set of 40. So you see somewhat distinct clustering, and this based upon color, based upon body site, and this is something that we have already known to be an important premise, an important guiding factor in understanding the nature of the human microbiome. The body site is most important. Your mouth is much more similar in terms of its microbial community to the mouth of the person next to you, even though you don't know them, than it is to your own gut. Okay? So body site trumps individual. But when we talk about one body site, now individual is an important source of variation. I'll talk a little bit about that. So we see the same thing during pregnancy. We also see this body site coherence play out over time. Now the x-axis is gestational day, and then subsequently postpartum day. Now, we look in particular at each body site alone because, as I just mentioned, this huge additional source of variation, site to site, means that we might miss some of the nuances and important detail that one would see when focusing on one site. So these are the data from just the vagina of that initial 40 subjects. We have close to 1,000 samples from these 40 subjects. This, again, they're weekly. And now, again, they're displayed against time. This is the x-axis. Now the color coding is based upon whether that woman delivered at term, that's in blue, or preterm, prior to the end of 37 weeks. Those samples are in purple. And what struck us was the fact that an unusually appearing large fraction of these samples from women that delivered early seem to be of a different compositional type. And the way that you see that here is because these purple samples seem to be higher on the x-axis, on the y-axis. The y-axis here is the score that came from the first component of that principal component analysis. And that score is an indirect and somewhat crude reflection of taxonomic composition. It had a higher score, a different composition. And the question was, what might be the nature of this somewhat distinct vaginal community that seems to appear early in pregnancy and be associated with those women who will subsequently deliver early? We looked at the structure of all of the vaginal communities and found something at first which was similar to studies that have been published of non-pregnant women, which is the existence of very discrete compositional states. In the vagina, they are numbered 1 to 5, and we think, in fact, that there may be probably a larger number in a broader population of women, but the literature and our data would support the existence of discrete types of a small number. The type that happened to be associated with that early gestational picture in women who delivered prematurely is very similar to what you might be able to see on these slides as the type 4 state. So it's in that little pinkish salmon color on that top bar that goes across the top. The columns that lie underneath the number 4 are the various taxa that make up the various samples that fall within class 4. They're little tiny columns within that segment. So what is a type 4 community? It turns out to be a community that's very distinct from all the other 4 types. It's the one type where you don't have a dominant lactobacillus species. In all other typical states of the vaginal microbiota, there is a dominant lactobacillus species, and that species may vary. Each woman here tends to have one of those types, and it tends to be maintained over time, although it's been shown quite clearly that the composition will change as a function of the menstrual cycle, for example. So we came into this study thinking, would in fact the absence of the menstrual cycle mean that there was now less variability over time, or might it mean that instead we have a different kind of dynamic of hormonal change that might cause there to be a clear effect of pregnancy of a more sustained stress-like disturbance type? So we first pursued this idea of type 4 association and looked at stability as well. And here are just some data that show that in fact type 4 is statistically significantly associated with preterm deliveries, and so we went to try to build a case for causation. So what do you do when you've got a putative causative factor, this atypical vaginal community, and you want to, in the future, prove causation? Well, you can't take this entity and introduce it into a new relevant host and reproduce the phenotype the way Robert Koch would have you do. And so we're now relying upon alternative postulates that have been established by epidemiologists for quite some time, looking at the role of cancer and environmental factors on the occurrence of cancer. So on the left you see what might be akin to a dose effect. This is something that epidemiologists have typically sought to establish as one feature of a causal argument. And without going into the details, again, this is published, there is some evidence of a dose-like effect. On the right is a temporal effect. Does this putative factor appear prior to the appearance of the endpoint of interest, premature labor? And in fact, it does. Now, these are just two arguments towards a body of would-be evidence that we would use to try to assert causation. But we know that causation of prematurity is almost certainly to be a multifactorial process. We don't seek or expect to explain a large fraction of it simply based upon an altered microbiota feature. And I've talked to you about taxonomic composition. We're busy now looking at the genomic composition of these communities, both vaginal and otherwise. We're looking at the metabolite composition of these samples from all of these body sites. And yet all of that may be only one piece of what is an interwoven, inextricably interconnected set of causal factors leading to premature labor and delivery. So this is still an investigation and a pursuit in progress. Many others are pursuing the same issue. I don't have the data here to show you, but this type 4 and other type community feature is relatively stable during the course of pregnancy compared to data from others in non-pregnant times. And we're collecting now direct comparison subjects for non-pregnancy states. But it raises a series of questions. How might this be related to prematurity, for example? Well, you might think about this history of aberrant assembly and now the establishment of an altered state which has properties that might be pro-inflammatory, because we know that a sustained or aberrant or improperly located inflammatory signal may be an important trigger for premature labor. Perhaps the problem is that these altered vaginal communities are now poor, impaired in their ability to do what normal human microbial communities do, which is to exclude invading pathogens. Maybe that's the issue. Maybe it's the absence of something, in fact, rather than the presence of something that's important. We know, and I just told you, that it's, in fact, the absence of lactobacillus that helps define the most gross feature of this altered community, type 4. Maybe it's an indirect effect. Maybe either a direct product, a metabolite, or a local immunologic aberrant signal is now transmitted into the systemic immune system or into the systemic circulation of this metabolite. Now maybe we're talking about an indirect route to the birth sac and to the tissues that are in charge of determining the onset of labor. This is something that we're pursuing and seeking to develop models. Now, I thought I'd just put up this paper because this is a paper that raised a great deal of public awareness and attention to the possibility that perhaps there are microbes themselves present before birth in the birth sac. And I just want to, without going into the detail, suggest that we're still in a position of relative uncertainty about what that might mean. For one thing, we don't know that those findings, nor our own prior, where we looked at amniotic fluid and found microbial sequence, means that there were, in fact, microbes, or are, in fact, microbes. And if there are, we don't know whether they are viable. But maybe viability is not the issue. Maybe there is a dormant state of these microbes that is nonetheless accompanied by subtle patterns of expression, of activity that is important. None of this is clear to us, nor in fact is the possibility that these sequences could be in part, if not entirely in some studies, contaminant sequences. This is not talking about low biomass samples like the placenta or like blood during health. So there are a number of issues that need to be sorted out, and I won't talk about these now unless there are questions later. I do want to just remind you, though, that the presence of a series of disparate microbes or their DNA does not constitute a community. The ecologists, getting back to this traditional discipline that I think has so much to offer our understanding and way of thinking, a community to them is much more than a collection. And I want to emphasize that. A community is all about interactions. It's about adaptation over long periods of time, short periods of time. It's defined in the relationship to its local environment, which is us in this particular case, and to a variety of processes that sometimes are difficult to measure. So think about this as we think about the microbiome and about our relationship to the microbial world in general. Postpartum, just one snippet from this published study, and we're pursuing this as well now. We found a very dramatic disturbance that takes place, at least in the vaginal microbial communities, right at the time of delivery. And this disturbance takes place in women who give birth to a child by both C-section or vaginal delivery. So it's not just the process of physical delivery through the birth canal, but the process of delivery itself. And you, of course, know well that there are, in either scenario, very distinct changes in hormonal environment. So maybe that's the issue, and yet it looks as though there's a clear time to restoration of what we might view as a more typical vaginal community, in this case, after delivery. You see these blue slopes with time, postpartum, trending back towards a state that I'm not showing you here, which is a more typical, normal vaginal community state. So what does that mean? To have a disturbance right at the time of delivery and a time to recovery. Well, it may mean nothing. Maybe this is just an epiphenomenon of looking at taxa, and perhaps we will find when we look at function or surrogates for function, metabolites and so forth, that there won't be such a dramatic disturbance. But I think there will be. And then we might ask, well, what is the functional consequence of this for the local environment of a now postpartum mom, as well as the systemic state of a postpartum mom? And could it be, this is just speculation, that the known linkage between interpartum period, the time to the next pregnancy, and the likelihood of a subsequent premature birth has to do with this reset? Maybe if you don't allow your microbiome to reset sufficiently, a new pregnancy now has an increased chance of going down this aberrant path or having a higher risk due to its microbiome disturbance. So, this is my last slide. I'm going to leave you with some summary statements and questions. I have sought to argue that this prenatal landscape is so important. There are lots of reasons for thinking about this, from those that study the developmental origins of health and disease in children